Today I'm going to talk about prevention of diabetes by knowing your genes. So as depicted here, the sequencing of the human genome offers the greatest opportunity for epidemiology since the John Snow discovered the Broad Street pump. And I would like to talk a little bit about what John Snow is, and he is the father of epidemiology. He was in the 1850s, I suppose, and there was an endemic of Corella in London City. And at that time, Corella bacteria was not found yet. But nevertheless, Dr. John Snow looked at the patients mapped on the London map and found out that those who were using this pump, which was actually contaminated with sewage, was the cause of the endemic. And he asked London to stop this using this pump, and the endemic just ceased. So in a similar sense, now we have a human genome map. This is just a map. It has information of 22,000 genes, but the function of each gene is not totally understood. Maybe 1/3 of the gene the function is known, but not for the other 2/3. But nevertheless, if we have this good map, we could use that for the prevention of human diseases. And that's what I'm going to talk about today. So after completion of the Human Genome Project, it was known that our genomes are 99.9% identical at the DNA letter level of GATC. It consists of like 3 billion letters of GATC. So the difference is 0.1%, and which sums up to 3 million letters of GATC. And these differences are most of these differences are called SNPs, which stands for single nucleotide polymorphisms. And the SNPs are mostly cause the difference of our appearance, a difference in our eyes, eye colors, hair colors, and also susceptibility to disease, and different reaction towards pharmacological drugs. So after this, many studies have started to decipher which single nucleotide polymorphism SNP is related to which kind of phenotype. And here's an ancient question from the Hippocrates era. I read it. It's far more important to know what person the disease has than what disease the person has. So you really have to look into the person, not the disease, because the person have the disease, and the disease is something within the person. Of course, I'll go into the next stage, but this is what Hippocrates said about more than 2,500 years ago. And now we have the information of the genome, the information of the 22,000 genes, as well as the difference of human with 3 million genetic markers called SNPs. So the first thing that was found is that there are monogenic diseases. These are very rare diseases, and versus there are multifactorial diseases. Today I'm going to talk about mostly diabetes, and which is most typical multifactorial diseases. But as soon as the human genome was deciphered, it was just began like this time in maybe mid-90s. The gene identified for monogenic diseases just grew up like this, and you cannot see this, but
CFAM caring for well-being.